Okay. Slides are visible?
Yep.
Yes, good. Yeah, I'd like to thank everybody for joining us today. It's my pleasure to introduce Dr. Ivan Rusin today, a friend and colleague. Been a great experience working with him in the world of tissue chips and microphysiological systems for a long number of years. I think you'll find from his presentation the breadth of that experience, both here in the United States and across the continents of others as well. Sits on many agencies and advisory boards, and just a breadth of information. And the whole purpose, the thing that struck me that prompted this particular roundtable was the announcement by the NIH earlier this spring in April that everyone saw, where they were trying to deprioritize, I guess, the use of animals is one way to look at it. And so there was an announcement, as you can see, that they're prioritizing technologies that reduce the number of use of animals. And I think what we're going to try to do today is talk a little bit about that, provide some background. And then I'm going to talk a little bit about our cores here that we have in Houston, but they're available to anyone in the state of Texas, really, that can reach us. Where I think you'll see some plans where some of the technologies and techniques that we use could be applicable under this new schema. That's what I'm hoping to convince you anyway. Just some bullet points that one can take away from that announcement, and just a couple that I wanted to do just to set the groundwork for what we're going to talk about today, and that's to invite and expect these what they're calling NAMs, which is the new alternative methods. Um, Again, human relevance, justification, fit for purpose is a key phrase that you'll find. Context of use. These are catchphrases that you can use as you start trying to incorporate these into your work plans. If you need to use animals, as it says, explicitly justify the necessity and minimum numbers. So these are all things that could impact our research as we move forward. Peer review study sections are going to be looking at these as well. So when you submit your proposals, they're going to be having ways of looking at that. And then in your budgets, you can incorporate, development of some of these tech platforms into your proposals as well. So how can you do that? Like, obviously, align your specific aims, validation subsection, so that actually a part of your proposal. Choose contract research organizations or cores, hint me, with experience in this area. That's what I want to try to convince you in my part of the presentation a little bit. You can budget for these, and then you can schedule an early meeting with the FDA if you're actually moving towards IND enabling studies. And then what are new approach methodologies, the NAMs? A lot of different ways, computational, tissue chips, other cell-based model systems that allow you to do scaling. And what we're trying to do in our program is to try to position ourselves that you can do some testing prior to going into animals. So it would be a way of reducing the number of animals in those studies. Normally, we think about cells and culture for these model systems, but the last bullet point there is that they do accept some non-mammalian models such as Drosophila and C. elegans as new alternative methods as well. Fantastic. Such a great resource. I hope you all, as you think about this, you'll be thinking about how to incorporate some of this information into your research in the future. And there's a lot to consider. One of the things that struck me, I guess, just in the last couple of years with the TextVal program, moving into some of these other more advanced commercial chips, is the upfront cost to running those and the amount of money you have to spend just in the equipment, not the chips themselves, but just the equipment sometimes to run those chips. So, there's a lot of hidden costs with those as well. What I'd like to share with you all is what we're doing here in Houston with the screening centers that we have established here through the Gulf Coast Consortia, to which everyone at any Gulf Coast Consortia institution has access to. And really, we don't limit our access to, so we're open to anyone, any academic, any biotech, any pharma. We don't care. I mean, our facility, our resources are available to all comers. So I just want to put that up front. But we've been taking more of a cell-based approach. We don't do too much in devices. We do a lot with cell culture and cell-based models. So that you'll see that from my slides. I'm just going to try to go through some of the resources that you have available here. And I think just try to keep in mind, as Yvonne had talked about, the initiative coming out of the NIH trying to reduce the number of animals. And I think from our perspective, what we would like to be able to do is provide you ways to look at your research and to try to make best decisions before you go into animals. You know, we're trying to position ourselves, try to, you know, sort of between discovery and in your in vivo model systems to try to take maybe the better molecules going forward, ones that have less risk, ones that have better efficacy. And so that's the approach that we've been taking. So let me share my screen again and try to get through my slides here. And they're visible again. Good. We're actually in Houston. We're located in the Helix Park, the TMC3 building. And like I said, the title is Translational Engines, how our core facilities can help you turn your ideas into impact. And so we're part of the, as I said, the Helix Park. This is sort of an artist rendering of that. We're in this building here. Um, it opened in 2023, and so we've been here just a little over 2 years. But we're sort of between the Texas Medical Center to the north of us there, and MD Anderson's and growing South Campus, which is below us and off the bottom of the screen. So just to orient you a little bit. We're part of the Gulf Coast Consortia. We're also part of what we've started calling the Gulf Coast Consortia ecosystem for drug discovery and development. And you can see I listed as many of the GCC related cores that have formed over the years, from most of this through CPRIT funding. So a lot of this has happened since 2010 when CPRIT came online. And you can see the breadth of it as it relates to the drug discovery pipeline. And there are facilities, and the ones in maroon are the ones that Texas A&M is involved with. So you can see where we are, but then I tried to include the ones from many of the other centers as well: Baylor College of Medicine, UT Austin, UT San Antonio, TSU for doing the PK/PD studies formulation. So you can see that we touch in all the different areas. And the nice thing about that is sort of what says up in the top corner is because we're all Gulf Coast Consortia members, there's easy access to all of these things. There's a memorandum of understanding between the universities. And so people can come in without all of the paperwork and disagreements that go through trying to get those completed. Sometimes that can take months. And it's really pretty open society. We're all like-minded, individuals, and we're all willing to work and trying to help everyone succeed, at least from the core side. But there are no barriers, and it allows us to move projects between sites as well. So, as your project matures, you might need the resources from a different core. And so it's easy enough to move them between cores. And we do that. I mean, UT San Antonio Health Science Center and UT Austin have both good, very medicinal chemist groups there, in more of the traditional sense of medicinal chemistry. And they've made molecules that for investigators at MD Anderson or Baylor, and then they've come to us to do testing. So it's easy enough to move those items around. So that, the barriers are just not there. And more recently, the Gulf Coast Consortia has gotten involved in commercialization too. The NIH-funded REACH program, research, evaluation, commercialization. These are for people that are interested in commercializing their research, and this is a way to do that. And they run cohorts of new investigators 2 or 3 times a year. They can correct me in the chat if they wish. But there's that, and the Accelerator for Cancer Therapeutics is up for renewal, I think, this year too, with CPRIT. So there are a number of opportunities available, regardless of where you might be in the pipeline. For ourselves, we started in 2006, and we've been continuously funded since 2011. We started with philanthropic money. We were doing traditional high-throughput screening. We do that now alone or in combination. As we got more into cancer research, we found that combination screens were more of a priority. So we've learned to do combination screens of any number of ways to do that. We provide access to collections of approved drugs if you're doing translational research, or if you're doing more mechanistic studies or doing target identification validation. We have informer sets. So these are molecules that are designed against specific targets, and we accumulate as much information about those as we can, including their off-target effects when they're known. The core has primarily focused on translational and personalized studies. You'll see as we go through the presentation, we're broadening and expanding the operations, and you'll see how we've been doing that. Traditionally, we were doing drug discovery in pretty much the traditional ways, target and sequence-based discovery, and where you might use a biochemical endpoint, a biophysical measurements. We're now ourselves adding, since we moved into our new building in 2023, we added the capacity to be able to do medicinal chemistry on premises. And we're also doing synthetic peptide chemistry as well. And we match that and marry it really with our phenotypic drug discovery efforts as well. And so we've been working not so much in 2D models anymore. There's still a lot of those going on, but we've been doing a lot of work in 3D models, and particularly spheroids and organoids, sometimes monocellular, sometimes multicellular, sometimes part of a microphysiological system, as Stephan was pointing out. We work traditionally with cell lines, but that's evolved over time too, and we're getting many more investigators bringing us patient-derived samples, cell lines, or cells derived from a xenograft, for example. And we're also, our data science program has really grown over the last 5 years or so, and we're doing drug discovery and development using generative AI, not only for lead op efficacy and tox profiling, but we're also doing it for de novo design as well. For the last time that we looked, period between 20 and 24, you can see that we've had some impact, number of papers, number of investigators and institutions served. And so this is kind of just a timeline. The only reason I put this in here is because it shows the evolution of the program over time. You know, we're doing traditional high throughput small molecule screens in 2006. I came out of the industry at that time to come back into academics, and that's what we were doing. I was running a drug screening group at a local pharmaceutical company, and that's what we were doing. We were servicing, again, the GCC community, and just doing that, mostly biochemical. And then you can see, just as we've been progressing, how we've added different things. We got our first automated microscopes in 2011, for example. These were ones that are just learned how to do an autofocus, for example. And so that became an important model. We started doing 3D model systems and you know, suspension cultures, things like that. In 15, 16, I got involved with Yvonne and Dr. Rusin in doing these MPS things. And I learned, you know, a lot about tissue chips from him and their group. They're just a phenomenal resource. Other things we've done, metabolic imaging. We have a unique way of looking at the metabolic profiling of cells, where we use the autofluorescence properties of NAD, AH, and FAD. And so in cancer, anyway, you can use these. It's non-lethal. It's label-free. We can do it in real time. And you can actually look at the cell's metabolic profile. You can see, are they using oxphos to generate energy, or are they using glycolysis to do that? And the idea behind that is can you find a vulnerability in the cells by maybe switching it? So if a cell becomes resistant to standard of care, perhaps it's changed its metabolic profile. And so now you need to combine it with something that might work with it under those circumstances. So it gave us a way to do that. And we started working a lot with organoids in 2020. And these are organoids that could be spheres that we can grow in ultra low attachment plates, where you plate the cells, they self-associate into an aggregate to form a sphere. We do a lot of work in cells and grown in gels. And so we have organoids that grow out from single cell suspensions in gels. Those cultures, depending on how fast they grow, we can keep those in culture for a week. And we've gone out almost as far as 8 weeks on some of those, just to try to allow these organoids to grow out. More recently, in 2021, is when we started doing what we call our MLOTS program, which I'll describe a little bit more. And the whole idea there was to do some de-risking, both in efficacy testing for the investigators that are in sort of in that lead op phase of molecular development. And de-risking as you're moving into your in vivo preclinical studies, or if you're thinking about moving towards IND enabling or clinical trials in your future, could you ask the question, do I have a risk, a liability in my molecules? And their whole purpose for doing that was helping investigators develop their portfolios. So if you have a company and you have a compound that you're trying to develop and you might want to out-license it at some time, well, you'd like to know perhaps if it has a tox liability in the heart or in the CNS or in the liver. Those are the three areas that we tend to focus on. And then our most recent funding that we just got in June of this year from CPRIT, we're doing probe discovery and supporting lead optimization still. So the first core I'd like to describe basically is our combinatorial drug discovery program. Again, this is where we are doing a lot of different studies where we're combining therapeutics in this case. We have a number of ways of doing that. Two that we do, and primarily is one's called anchor and probe, where you have like a standard of care, for example, that is your anchor molecule. And then you've got resistant cell lines and you're trying to find ways that you can combine standard of care with something else in order to be effective in the realm of cancer. Now, you can apply this to anything. I mean, we're CPRIT funded for 75% of what we do. So a lot of my references are going to be to cancer, but that doesn't mean that they don't apply to any other system where you might want to combine agents, and we can do that. That might be more of a pairwise combination, or three things or four things. We can do those kinds of combinations. Most of our screens are done in multi-well plates. We work with a lot of automation, so we don't have a lot of headcount, but we do a lot of projects because of the automation. It allows us to have the throughput that we're looking for. And then our endpoint detectors can be microscopic in ways of doing that. If you have reporter systems, we can do flow cytometry, again, optical metabolic imaging. So just about anything that you can think about from a biochemical standpoint, absorbance, UV-Vis, just about any form of fluorescence endpoint, alpha screen. We have flow cytometry. We have both analytical flow and we have spectral flow. So if you're doing biomarker panels where you might have 20, 30 markers, our spectral flow might be a way of doing that. And again, I just touched again on optical metabolic imaging. Our newest core, funded by CPRIT, is the Drug Discovery Resource Center. And this is where we added the medicinal chemistry and really built out the ability to apply data science to the generation of new molecules. And that would be our AI-enabled cheminformatics. I'm using it in the MLOTS program for predictive toxicology, sort of the PBBK QSP approach to doing that. We expanded our screening capabilities. We added the ability to do biophysical kinetic measurements, for example. We brought back online surface plasmon resonance and isothermal calorimetry when we brought a very seasoned drug discovery expert in from St. Jude's earlier this year. And then we're doing small molecules, but we're also doing peptides, synthetic molecules. We're interested primarily in macrocyclic molecules with and without non-canonical amino acids. A lot of these compounds are starting to take on drug-like properties. It's something we're interested in developing, perhaps as shuttle vectors in the future. And then to support our AI, our data science program, we have an iterative way of doing that. So, Yvonne spoke about making models. So PBBK is one way of doing that. But a lot of times you see these studies, but they're all done retrospectively. The data has already been collected. They go back and they analyze and they develop a model. What we want to do is actually do the prospective, but have a human in loop and then a feedback mechanism to take the data that we generate, feedback, and improve the model systems. And so we're hoping to do that through our program as well. So again, the Drug Discovery Resource Center has the ability of doing design, make, test, and analyze, the DMTA. And you can see this is just a list of a lot of the technologies that we're applying in the lab to those. I understand the slides will be available, so you don't have to gather it all, but the information will be there for you to look at. And then they're being applied to some actual projects. So we're just getting started in those, but you can see there's a sort of a breadth of target areas that we can apply this to. And so we're working it through and we're working towards moving these compounds further into discovery. And then MLOTS is the last one that I'd like to proposed, and that's our microphysiological lead optimization system. And the reason we call it the microphysiologicals, at the time, I was working with Yvonne, and it was a good fit because we were thinking about like momentous tissue chips. We were talking about blood-brain barrier that we had done with them. So those types of model systems that were fairly straightforward, but then starting to be predictive. And we were trying to do them in human cells. So again, it sort of fits with the NIH initiative of where we were trying to do more human type studies, so moving away perhaps from animals, use of animal cell lines, but then moving towards where we could, iPS-derived human cell lines and the like. And so there we've been working with the spheroids, organoids. We've got some applications. I'll show one example where we're doing adaptive cell therapeutics using these types of model systems as well. For our neuro and cardiotox methods, we use microelectrode array reader technology, where we can actually look at the electrophysiological responses of those cells in culture, and we're using iPS-derived human cardiomyocytes and neurons. We also have the ability to do impedance, so we can do real-time measurements of cell growth and cytotoxicity, those types of measurements. And the output in our case is we're trying to consider drugs that are moving towards preclinical, perhaps maybe even preclinical IND enabling studies, and saying, if you had to make a choice, you obviously can't take 5 or 6 compounds forward into an animal study. It might be too big or too expensive. Could you make choices based on information that you're gathering to make the best choice of 1, 2 of those molecules, maybe instead of all 6? So we think there's some translational value from that as well. So as I pointed out, we're trying to put MLOT sort of as a bridge between the new drug discovery as you're moving towards lead identification, lead optimization, and you're moving towards your IND enabling studies, trying to make best decisions based on data. So, you have go, no-go decisions about whether you want to go forward into those studies. Well, you need data to make those decisions. And that's where we're hoping that we're providing that kind of information. We think of it as a fail early approach, because really what you would like to do is, if you have a drug that has a severe liability, you'd probably like to know that before you go into those kinds of very expensive studies. And so that's the idea behind that, is to try to figure that out early. And if it's early in lead up, maybe you can go back and do more medicinal chemistry, some additional SAR, maybe engineer it out. So that there are a number of opportunities of ways to move forward with that. And again, some of the different ways that we've been doing things, we have a very good, my colleague, Shreya Raghavan in College Station, Department of Biomedical Engineering, part of TEAS there, has developed a proprietary hanging drop rack, 384 well, the best one that I've worked with, because it works with automation, which is fantastic. But you can see in the picture there, it doesn't take many cells, and she's worked with as, for those of you that have very rare cell lines, she's started with as few as 10 cells in a well and can watch organoids grow out from those. The organoids you can see come in all different flavors. Up the top right corner there, you can just see an example. They can be nice and smooth and round, and you can see what's going on, or they can turn all kinds of spiky and take all kinds of different kinds of phenotypes. And so you can monitor that. We can watch those in real time. That's the beauty of these kinds of model systems, is that you can add a temporal component to the whole thing. So it's not just an endpoint assay. A lot of these are going to have that temporal component. You can see what's going on in real time. The momentous chips, as Dr. Rusin measured, our BBB model, then our lead op profiling models as well. And we also do a liver tox, but that's a high content microscopy method as well. So again, this is just sort of a list of the things that are going on in the lab, not necessarily anything that you need to really look at, but just it's a reference. I hope that you'll look at the slides or the recordings later on. You can see maybe these are some model systems and methods and technologies that might be applicable to the research that you're doing. Like I said, we do a lot because of our CPRIT funding in cancer, but again, we're part of the Gulf Coast Consortium, which means we can work on just about anything that comes in. And that's always been the philosophy of the core, is can we be flexible? And so you can see when we start looking at what we're capable of doing, we can work on anything from small molecules to cell-based therapies. We do have an X-ray generator in the lab as well. So some of the work that we've done in cancer, but we also have had some NASA projects come through that have been real fun, where they use high high radiation exposure. And so we have an X-ray generator on premises, and allows us to do those kinds of studies. And so we can work with just about any type of molecule. And then model systems, we've worked with a lot of different model systems, as you can see, anything from a purified protein in a true high throughput fashion, as we used to do, to some of the more complex model systems. And we've done a little bit of work with model organisms as well. So, those are all capable, and they all have, as we said early on, a context of use, and they all have to, you find the right model that's fit for purpose. That was a sort of a phrase that is important, that you're using the right model to ask the question or answer the questions that you're trying to ask. As Dr. Rusin pointed out, not all models are suitable to answer all questions. And so it's a matter of trying to find the right one. Again, we use automation. We have lots of ways to assemble assays and bring those things together. And we've brought that equipment together for you, including access to libraries. I mean, the drug libraries themselves is a nice resource. I mean, even if we may not have the ability to do your assay, we can still provide you with compounds as well. So that's something to think about if you need access to compounds in a library. The libraries are expensive. If you wanted to go out and buy one for yourself, you might spend $10,000 or $20,000 on a library of a few thousand compounds that you won't use one or twice. And that way, that's not very cost effective. There's no value proposition in that for you. But using us as a resource, you can have access to our compounds. You pay for just what you use. And so, it's cost amortized over everyone that's using them, and it's very good. And we have a host of readout devices, as I said, anything from biochemical assays, and you kind of get an idea of the flow and the throughput that we can do. And these are all per day values for the different models that we do. Biochemical assays that are high throughput imaging. We can do some high content imaging as well. We have automated, our flow cytometry is automated. We can run unattended for more than 24 hours. So those of you who have done flow by tubes in the past, a few tubes, 40, 50 tubes a day is a long day. But, for us, we can do somewhere around 8,000 wells a day from sampling from a 384 well plate. Our microelectrode arrays, again, we can work on, the electrophysiology of neurons and cardiomyocytes, but we can also look at scratch assays, for example, or anything with cell growth, because it uses, the impedance is actually transepithelial resistance, so it's a tear measurement. And so we can look and see what happens with monolayers of cells that grow on the electrodes. And then again, our biophysical measurements as well as surface plasmon resonance, isothermal calorimetry. And I wanted to just give the flavor of one example. And the reason I did this one in particular is because it uses a lot of the techniques in the lab. There was a group that Katie Rizvani and her group came to us about a year ago and said, do you have a way that we could look to see if we could find some way to prolong the activity of our CAR NK cells? Could we treat them ex vivo and sort of energize them and then treat tumor cells with them to see if they would last longer, make them more efficacious? And so we devised a platform for doing that. And the approach that we're using is the question we're asking is, can we get CAR NK cells treated with compounds to change them to make them more robust, make them work better against a solid tumor? In this case, it's a spheroid. And so we take tumor cells that are expressing GFP. We can plate those in ultra-low attachment bottom plates, and you can see that they form a very nice spheroid, which is dependent on the number of cells placed in the well. These cells self-assemble into a spheroid. And so we grow those for about 2 days. The NK cells are prepared, the CAR NK cells are prepared by the research group there at MD Anderson, and then we bring them together. And so we use an automated transfer station, as you can see in the bottom there, and we transfer our spheroids on top of the CAR NK cells. And then we ask, can those cells kill the tumor? And what we're doing is we're using flow cytometry to see when we drug treat the NK cells, that do we or do we not have drug effects on the NKs themselves? So obviously we don't want cytotoxic chemicals on those. So we're only looking for compounds that don't kill the CAR NK cells. And then we marry those with the spheroids. And so then we use imaging as an endpoint for that. to see what happens to our spheroids. And since the cells are labeled with GFP, we have a beautiful reporter, and we're doing time series analysis on that. And so about every hour, hour and a half, we collect images and we can see what goes on with those cells over time. And then just as an orthogonal, we run cell titer glow, so we can do that as well. So we did assay development with them where we developed this method that you see before you. The analysis algorithms, how are we going to image how do we analyze the flow data? How do we analyze the imaging data? Can we use automation? It was important to know that we could actually scale this down to a 384 well plate. We did pilot screens to test all of those, and then we developed orthogonal assays. And then we build in rigor and reproducibility. So biological replicates where we can do them, obviously. Technical replicates in the assay if our Z primes are really below 0.4. These ones are actually fairly reproducible. Our Z primes have been running about 445.5, so pretty good assay for us. And then every plate that we run has on-plate controls, and that's just part of what's built into all these assays and screens that we do. Obviously vehicle controls, but then we also have known controls on there for our C prime. And another statistic that we calculate called minimum significant ratio, where you run full dose response curves and you're actually measuring how much difference you see in your IC50s over time. So this is just a real quick representation of the data. If you do just let the tumor cells grow in culture, you can see over time, you can see an increase in the intensity of the GFP signal. So the cells don't necessarily get bigger, but they get more dense. And so we can see more green. If you treat the cells with a drug, in this case, I'm sorry, I can't give you the name of the drug, but drug A. The dose response goes from the black is untreated, from brown, lowest concentration, to red at the highest concentration. So you can see, as you can see in the concentration response curve to the right, increasing concentrations of that had a negative impact on the NK cells, but it also seemed to have an effect on the tumor cells as well at very high concentrations. So just reorganize those three graphs a little bit there to the left and bring in the new graphs. If you have the tumor cells plus the NK cells, you can see that the NK cells are killing the tumor cells. And you can see that in the top, the 0.0, those are NK cells, just NK cells with tumor cells. And so you can see over time in the right-hand panel, you see the spheroid disappearing in time. So the NK cells are killing the tumor cells. If we treat the NK cells with drug first, and then add them to the tumor cells or add the tumor cells to those, you can see the black line is that NK tumor only, but then you see the two brown lines to the left of those, and you can see that the killing was more rapid. And you'll notice that those are the brown lines, which are the two lowest concentrations of that drug. So the -9 and the -9.5. And you can see the -9.5 is illustrated in the video in the middle. You can see that the spheroid is disappearing in time, and they're sort of time matched. You can see that it goes away much quicker than the panel right above it. But if we go too high in the concentration, so if we go out to 10 micromolar -5, then you can see that we were killing the NK cells, right, at -5, that bottom blue graph, the CAR NK alone plus drug. If you get it and look at -5, we've killed just about all the cells. And you can see when you look at the -5 treated there at the bottom panel, those cells don't go away and the spheroid just stays. And so it kind of goes along with that. So back to the NIH and their initiative. So one of the things that we're going to be watching very closely is the standardized organoid modeling center. And these are just some of the points that are made out of that. But the biggest thing is in the top in the red, it's going to be a hub for standardization, developing organoids that are reproducible, reliable, easily accessible for medicinal biological reference. And so they're going to be establishing protocols. So we're going to be watching as a group that does organoid work. We're going to be watching this very carefully. We're also involved in AI and ML, and so we're going to be watching to see how that goes too. And if they're going to make all of this available to the community, obviously we need to take advantage of it. And so that's what we're going to be doing. They're promising to have affordable access. It says it's available at minimum cost. So they don't actually state what that is. NIH funded and other qualified investigators. And again, there's no definition as to what those things are at this time based on the press releases that they did. but they're also going to do, validated using standards and those that are implied, for FDA and OECD and ICMM. So, it's going to be something worth watching. And I think we're going to see over time, how that impacts our research. Different ways to interact with us. We're very collaborative. We can work strictly on a fee-for-service basis, especially for those of you who are on grants where the budgets are really tight. You can just pay for what you use. We've worked as an honest broker between, say, the pharmaceutical company, which has drugs for testing, and a model system that you're developing in your lab, but maybe neither one wants to give it to the other. You can bring it to us. We're more than happy to automate the screening and share the results with everybody. And we're happy to work blinded on those things. And naturally, we can work on a contract basis as well. So this is just a repeat of what I said before, and time is short. So I'm just going to skip that and say thank you. This is the leadership here. Our funding is mostly from the Cancer Prevention Research Institute in Texas, CPRIT, but we also have some NIH funding in our fellowships. This is what funds us. The QR code will take you to our GitHub page where you can find all the information I shared with you today and much more. And so I'm going to stop there, and I think we're ready to take on some questions and have a nice discussion with everyone else. Stop share. There we go. So